This is part 4 of a series of videos describing how to perform ChIP-seq analysis using Galaxy. The main aim of this part is to discuss how the so-called peak calling step of ChIP-seq analysis works. The most widely used peak caller is MACS, which stands for Model-Based Analysis of ChIP-seq, and basically identifies regions of the genome with a greater abundance of enriched ChIP reads compared to input reads. One of the main reasons for making this new series is because there is now a new version of MACS, called MACS2, that was released well over a year ago. At this time, MACS2 is at version 2.1.0. If you have previously used MACS version 1, a few things have changed. Coverage plots are now in bigwig format instead of wig. This is important as bigwig is a smaller compressed version that measures fragment coverage in ranges rather than each base individually. The empirical false discovery rate, FDR, has been replaced by the Benjamini-Hochberg qvalue. And finally, there are also changes to the default settings, which means that some of the parameters now have different settings, so you can't necessarily rely on old command lines. Now, our group has released a MACS2 tool on the Galaxy toolshed, and it's been tweaked to basically suit our requirements, but it's available for anyone to use. And if you're looking for this on the Galaxy toolshed, the author is P.J. Briggs. So in this part, I'm going to discuss the parameters and concepts that are used in MACS2 peak calling. I'm also going to show how to use MACS2 in Galaxy and show some of the output files. And finally, show you how you can simply visualize the results using a standalone genome browser called IGB. The project home page is at this web page here, and all the development and new versions of the software can be found at the project's GitHub page. This is just to remind you that it's P.J. Briggs you need to look for as the author when looking for the tool on the Galaxy toolshed. So now I'm just going to take some time just to go over some of the parameters and concepts used in MACS2. Now I may have mentioned this before, but it can't be repeated enough, is the need for a background or DNA input sample. So a typical control for a ChIP-seq peak calling experiment is DNA input, and this is usually DNA split from the sample that has been prepared for the chromatin immunoprecipitation procedure. But this split piece of sample does not undergo enrichment using the ChIP antibody. Read coverage over the genome isn't uniform, and the peak caller uses the input reads to identify spikes of read coverage that are artifactual due to the underlying sequence. For example, the amplification step during the sequence library preparation can create sequence-specific amplification artifacts, which basically on a genome browser look like real peaks. In a single-end read mapping, only one end of the DNA fragment is actually sequenced. Therefore, it is not known how long each DNA fragment captured by the immunoprecipitation was. This information is required to accurately create the pile-up of fragments and calculate the position of the summit, or the highest part of the peak. And this is most likely to be where a transcription factor binds to the chromatin. MACS2 now uses a method called strand cross-correlation that relies on forward and reverse reads clustering in areas of binding. When paired end reads are used, the length of the DNA fragments are actually known. Simply put, MACS2 is looking for regions of the genome where chip fragments are clustered more than the input fragments are in the same region. Of course, nothing's ever simple, and the authors have tried to model in the fact that the genome is not homogeneous and that some areas will be bound greater than others. So if cross-correlation fails, you can opt not to actually build a model and specify your own fragment size. So I'm now going to try and describe how MACS2 attributes significance to the different candidate binding regions. Now, significance is determined using a Poisson test, which uses a local lambda model or a background model. Now, this is quite complex, but I've tried to simplify it. And so for each candidate region, the IP, the chip fragment coverage is determined and divided by the region length. So this is taken and then compared to the local lambda, and this background model is comes from a number of different aspects of the genome. So firstly, the input reads in the candidate binding region, that coverage is also determined, and that's one facet of the background model. The coverage over the entire genome background is also calculated. And then, to try and take into account local fluctuations and biases caused by the actual sequence itself, windows of sequence centered upon the candidate binding region of 1 kilobase, 5 kilobase and 10 kilobases, they're also taken and the input coverage is determined. And all these factors go into a big black box, and the background facet with the largest region coverage is compared against the input fragment coverage, and using the Poisson test, significance is attributed to each binding region. The Poisson statistic is the probability of observing the reads in the candidate region with lambda as its expectation. Now, although MACS2 can be run only using a chip sample, it is best to include a DNA input sample that matches that chip sample, as I discussed earlier, and in doing so removes false positive binding regions from the downstream analysis. So basically, if you don't use a background input sample, you can't construct a background lambda model for the statistical significance test. The Bonferroni-Hochberg qvalue replaces the previous empirical FDR value, false discovery rate. Both methods, though, are a way of controlling for multiple testing in the same experiment. Very simply put, if you had tens of thousands of binding regions, each with a p-value cutoff for deciding whether it's a true peak or not, a p-value in itself is saying that there's a certain chance that an experiment could be a false positive. So the FDR value, or now is used the qvalue, attempt to correct for this. So by default the qvalue is set at 0.01 and is used as the significant threshold for reporting binding regions. A slightly more lenient 0.05 value is suggested for more diffuse binding regions such as histone marks. I will then sort the binding regions by the fold enrichment value, which is found in the detailed spreadsheet of results, but I'll explain that later. And the fold enrichment value compares the chip fragment count in the binding region against the local background or lambda model. The effective genome size defines the mappable or usable area of the genome. For example, highly repetitive sequence is difficult or impossible to map accurately, which leaves about 70 to 90% of the genome mappable, depending on the organism. MaxTo provides defaults for widely used organisms, such as human, mouse, fly, etc. This value is used to modify the lambda calculation and is used when allowing MaxTo to decide how many duplicate or redundant reads can be used. I'm going to describe that concept later. Now, the bedGraph file, which replaces the old WIG file, is useful to export fragment coverage data. They can be displayed on the UCSC genome browser or other genome browser, showing a profile of peaks and troughs over the genome for both the chip sample and the calculated background model that is based on the input reads. Other browsers are available, such as Ensemble, the Integrative Genome Viewer, or the Integrated Genome Browser. A useful feature associated with the bedGraph output is the signal per million reads setting, as it normalizes the coverage score to the n million chip reads used when finding the peaks. This means that the tracks can be compared between samples and experiments, which is useful when the total read counts are very different. So basically, for each range in the bedGraph file, the value is multiplied by, for example, 10 million reads divided by a million. The bandwidth is only used for fragment length cross-correlation when single-end reads are used. It is defined as the mean DNA fragment length as observed on a gel prior to library preparation. So this is set by the user. I just generally use a value of 300 base pairs, as the smear on a gel generally centres on this position, and to be honest doesn't have a great effect on the general outcome. The MFold value, well, in the advanced option of the Galaxy tool you may wish to alter the MFold value that changes the number of test peaks used in the cross-correlation phase when single-end reads are used. Now, setting this too high or too low can affect the number of test peaks that are used to estimate the fragment size, but generally speaking 10 to 30 is okay. The Cool Summits setting allows the detection of subpeaks when you believe that there may be multiple binding events in one cooled region. This can often be seen as multiple peaks appearing in one candidate binding region. So setting this will give you potentially multiple peaks per binding region. Now this is useful because if you suspect there to be many binding regions clustered together, this potentially allows you to separate those out, whereas before you'd only focus on the one summit in a binding region. The only thing to be careful about is that in the later stages where you want to extract sequence around a summit, or in this case separate subpeaks, you need to make sure that the sequences don't overlap. So lastly, I'm just going to mention about the concept of non-redundant reads in MACS2. So the peak caller uses non-redundant and extended reads, which makes a fragment. Reads are removed that share the same strand and 5' end position by default, only leaving one example. This aids in removing artifacts caused by the sequence, for example regions that cause PCR overamplification during the library preparation step. Now, when paired end fragments are used, it is required that either the 5' or 3' end of that fragment be different. Now you can also set MACS2 to allow some redundancy that can be automatically determined according to the effective genome size and the number of reads, or you can set the level of redundancy yourself. Okay, so now I'm going to describe how to run MACS2 in Galaxy, and just to remind you, this is using our own tool that was designed by our core facility. So the input in this case for MACS2 are the two bed files containing the trimmed reads that were mapped in a single-ended fashion to MM9, the reference genome, and were then filtered to only retain those reads that belong to the main chromosomes. So for mouse, that's chromosome 1 to 19, X and Y. So to find MACS2, we can type that into the toolbar on the left, select MACS, and then we're presented with all the different options for the parameters. Okay, so like I said, there are many of the parameters that you don't need to touch, but so the first one is you can give it a more meaningful name, perhaps containing the name of the factor and the genome. You then have to decide what type of binding region that you're looking for, or that suits your experiment. Now, narrow regions are most suitable for transcription factors and histone marks that have punctate or very peak-like profiles. The alternative are broad regions that are seen in some histone marks that cover very large regions of the genome. And when this value is selected, it has its own p-value cutoff. This is because the broad regions tend to have less fold enrichment. You have to obviously provide the tool with the input reads. So in our case, that is the filtered chip reads and the filtered input reads. Just find those there. The effective genome size has a number of predefined values. So we'd choose mouse, but there's also entries for human, worm and fruit fly. You can also input your own value if you're using a genome that's not covered here. Bandwidth we can leave alone. So include XLS file from MACS. We need this in the output because this contains all the in-depth statistical information that allow us to threshold and subselect the binding regions that are given to us. We also want to retain the bedGraph files because these are required in downstream analyses. It's always good to have them anyway, unless perhaps you're just having a quick run of the tool to get an idea of how many binding regions there are. Now the binding regions can be selected based on the qvalue, which is set at 0.01, but you can also use the p-value if you have reason to do this. We've got the advanced options that I'll come to in a second, but the build model option is applicable to when you are using single-ended reads and you need to determine the fragment size. Now sometimes this may fail for various reasons, such as there aren't enough test peaks to make an estimation, and you can turn this model off by selecting --, no model enabled. So let's look at the advanced options. The first two are the MFold values, and this is to do with the cross-correlation model prediction. And then we have options to turn off the dynamic lambda model. Detect subpeaks. This is to do with if you want to find subpeaks within called binding regions. So, like I said, sometimes you can find that the binding regions determined by MACS contain two or more discrete peaks, which might be discrete binding events. Now, this last one, use of duplicate reads, this is to do with how many redundant reads in one position you will allow to be used. Now, by default this is 1, this is the author's recommendation. However, you can increase this number, or you can allow all duplicates, or you can allow MACS to determine this number automatically. MaxTo is able to use a negative binomial model which takes into account the size of the genome based on the effective genome size you provided and the total read coverage. So when you are happy with all your parameter choices, you can then click on execute and allow Galaxy to run the analysis for you. Now next I'm going to describe briefly the output files that are produced by MACS. Okay, so the first file that we could look at is the HTML report. Any of these files can be looked at with the eye icon that brings the details up in the middle screen. This is basically a log file that shows you all the output of the program as it runs, and is useful to keep for your records. One thing that's interesting to look at the top is a PDF file which shows the how the fragment length model prediction performed. This plot here shows that the highest peak is the greatest or most prevalent distance between the test peaks, so in this case 113 bases. You'll see that there are other predictions corresponding to other peaks or shoulders on the plot. So there isn't actually anything else on this log file that isn't in the other files, but still it's useful to have for your records. So the next file to consider is the SummitsBed file, and this contains the coordinates of the summit of every peak. So this is the highest point of the fragment pile-up in every binding region. So we've got chromosome start end, a name given by MACS2 and the corresponding qvalue. Now if you had selected call summits and got the subpeaks reported, then you would receive multiple summits with A, B, C, D after their names to show that they belong to the same peak, same binding region. So moving on, the next peak is in this case the narrow peak file, but if you're running the broad version of the analysis, this would be a broad peak. and if we look at this, this is an encode format, and it's particularly useful in that it will display both the start and end of the entire candidate region plus the summit in one track. There's nothing else we need to do with this file in particular, apart that it is useful to display on genome browsers such as the UCSC browser. So probably the most important and useful of the output files is the peaks or interval file. Now in essence this is a tab delimited text file, but this can easily be loaded into programs such as Excel or LibreOffice Calc as a spreadsheet. Now what I tend to do is put all these items at the top into a separate sheet and keep all these on another, because then you can easily sort by the different columns. Now all these pieces of information at the top basically tell you what all the settings you used were. Gives you D, which is the estimated fragment size, and its alternatives. So you have the chromosome start and end of the whole of each binding region that was found. You have its length, the coordinate of the summit for that binding region, the number of chip reads in the pile-up, its p-value, fold enrichment, which is the fold enrichment of the chip reads over the background model, and the corresponding q-value. When sorting the file, I sort by p-value, not q-value, but I use the q-value as the threshold statistic. The reason I sort by p-value is these tend to be discrete values, where you'll sometimes see that the q-value step so that you get the same value, and this isn't good when you're sorting. So sort by p-value. But then also what I do is I sort by fold enrichment score. Now the reason is because as you go down the file, the fold enrichment score will drop, and I tend to consider anything below a fold enrichment of 5 to be of slightly dubious quality, and this becomes clearer when you put the coverage files into a genome browser. So this file can be saved to your computer and converted into a spreadsheet, and then you can use other tools to add for example, the closest genes to each binding region, and annotate it however you like. Lastly, we have the two bedGraph files that show the coverage of the treatment or chip fragments in this case, and the control lambda background model based on the input reads. Now, for our and your convenience, we've also produced a bigwig version of the chip read coverage bedGraph. A bigwig is a indexed binary version of a bedGraph file that can be used in downstream analysis, and will actually be used in this tutorial in another program. I've mentioned several times about viewing and using the data from MaxTo, but just to summarise, the spreadsheet output is the most important of MACS2 as it allows you to filter the binding regions, create different genomic ranges for downstream analysis, and annotate with additional information. I always add the 2 closest genes to each binding region using a tool we developed called RNA chip integrator, which I will describe in a future video. It is also helpful to see the peaks, the coverage plots, on the genome. I generally use the UCSC genome browser to do this, as sessions or track hubs can be shared with people who use our services. These methods do require access to a web server, although people have apparently used Dropbox or other cloud services with differing success. For this tutorial I am recommending the integrated genome browser as it allows you to view the bedGraph files locally on your own computer. The IGB website contains all you need to get started, and their YouTube video does an excellent job of describing its functionality in relation to a ChIP-seq project. Another alternative is the Integrative Genome Viewer, IGV. That concludes part 4, and thank you for watching. Goodbye.